Assalamualaikum warahmatullahi wabarakatuh. In here, Group 3 will present the important role and benefits of analytical chemistry in everyday life. We choose the topic of the important role of analytical chemistry in medicine. But before that, we will introduce ourselves first. Starting from me, I am Zulfa Tadira Fatila with name M06240018. In today's modern life, medicines are an inseparable part of efforts to maintain and restore health. Medicines are one of the essential needs in the world of health. Every medicine consumed must be of good quality, safe to use, and provide therapeutic effect according to its indication. Have you ever thought, how can a medicine be declared safe for consumption? Is the dosage correct? Is the medicine free from harmful substances? To answer all these aspects, a science is needed that plays a role in identifying, measuring, and ensuring the quality of materials, namely analytical chemistry. Analytical chemistry is a branch of chemistry that focuses on the identification, characterization, and quantification of chemical substances in a sample. In the pharmaceutical world, the role of analytical chemistry is very important because it determines the safety, effectiveness, and quality of every drug product on the market. In the context of medicine, analytical chemistry is used to study the chemical composition of raw materials, active substances to the final product. This science is the foundation in the process of developing new drugs, quality control, and evaluating the stability of pharmaceutical products. Analytical chemistry is present as the main tool to ensure the drugs consumed by the public are free from contaminants, have the appropriate levels of active substances, and are stable during the shelf life. Without analytical chemistry, we will not be able to know whether a tablet contains the right dose of active substances, whether the drug is free from dangerous contaminants, or whether the drug is still suitable for use until its expiration date. Analytical chemistry is also a determinant in making important decisions such as stopping drug distribution, withdrawing product from the market, or improving drug formula. The following is an explanation of the point regarding the role and benefits of analytical chemistry is the field of medicine. 1. Drug discovery and development. Analytical chemistry plays a key role in the early stage of drug discovery and development. This process begins when researchers search for new chemical compounds that have the potential to act as active agents to treat a particular disease. In this stage, analytical chemistry is used to identify the structure of compounds, assess their purity, and measure their concentration. Techniques used such as NMR spectroscopy, UV spectrophotometry, gas chromatography, and high-performance liquid chromatography or HPLC are essential in determining the characteristics of compounds. Without analytical chemistry, it is difficult for scientists to determine whether a compound has potential as a drug, or whether it is safe to proceed to further testing stage such as preclinical and clinical trials. 2. Quality control and quality assurance. Chemical analysis is essential in quality control and assurance of manufactured drugs. It ensures the active ingredient levels are within standard limits and detects contaminants like heavy metals or residual solvents. Techniques such as HPLC, titration, and mass spectrometry are routinely used to guarantee that drugs are safe, effective, and consistent in quality. 3. Regulatory compliance. Every drug in circulation must obtain permission from a drug regulatory agency, such as BPOM in Indonesian, FDA in the United States or EMA in Europe. To obtain this distribution permit, pharmaceutical companies are required to include complete data on the result of chemical analysis of their products. This data includes specification of raw materials, levels of active substances, purity, stability tests, and validated analysis methods. Analytical chemistry is the main tool to ensure that pharmaceutical products meet international standards and do not harm consumers. The role of analytical chemistry is also closely related to legal aspect, regulation, and the industry's responsibility to society.
Hello, my name is Demistrianda from student number 25. After hearing the explanation about chemical analytic, we will talk about a case in pharmaceutical industry. The case is using analytical chemistry to detect falsified or counterfeit medical product. The case introduction will be explained by my friend Masako Kireina Anasis.
My name is Masako Kireina Anasis with name M0624011. Have you ever heard about counterfeit or falsified medicine? So, the counterfeiting of pharmaceutical products has become an increasingly serious issue over the past decades. According to the World Health Organization, falsified medical products are those that deliberately or fraudulently misrepresent their identity, composition, or source. Among the most commonly targeted drugs are those that affecting the central nervous system, such as psychotropic medications. While many counterfeit products lack active ingredients or contain incorrect ones, some are chemically similar to genuine drugs. Despite appearing to be accurate imitations, these counterfeits still violate the intellectual property rights and pose health risks to the poor manufacturing and storage conditions. Image of pressing machines confiscated from counterfeit production sites reveal clear neglect of good manufacturing practice or GMP standards, basic hygiene, and sterility protocols. Furthermore, the more closely a counterfeit drug mimics the original, the more challenging it becomes to authenticate. Therefore, a good analytical strategy is essential. Ultimately, chemical analysis plays a critical role in identifying counterfeit drugs, such as high-performance liquid chromatography, 3D microscopy, near-infrared spectroscopy, imaging tools, gas chromatography coupled with mass spectrometry, nuclear magnetic resonance, and X-ray fluorescence spectroscopy.
Hi, it's Janice again. Before jumping to the discussion, we will talk about what instrument they use in their research. So, in this research, they use HLPC, near spectroscopy, nuclear magnetic resonance, 3D microscope, X-ray fluorescence spectroscopy, and then the last one is the gas chromatography coupled with mass spectrometry or GCMS. Then, let's discuss. The evaluated imaging tools, spectral imaging, in-IR imaging, and 3D microscopy help consider physical difference between the genuine tablets and the counterfeits. The embossing, the glowing, the granulation, and the repetition of the compounds, like the active ingredients, could be detected as different in the counterfeit in comparison with the reference. In-IR imaging enables the counterfeit tablet analysis, the detailed compositional information, and the comparison without sample preparation with the reference, gathering both chemical and physical dimensions. Various spectroscopic techniques, including NMR, NIR, XRF, and GCMS, were effectively used to detect difference in the overall formulation of counterfeit tablets, particularly in excipients. NIR revealed slight variation in the OH bands, indicating a different sugar composition, which was later confirmed by GCMS. NIR also proved to be a reliable method for analyzing sugar content in the tablets. Additionally, differences in the levels of palmitic and stearic acid were detected by both NMR and GCMS, correlating with varying magnesium levels is identified through XRF. GCMS could be also applied to detect residual solvents in the tablets.
Hello, I'm Dia Alfatira, student number M0624003. I would like to continue the discussion from my friend Demis about HPLC and Raman spectroscopy. HPLC can be used to detect the impurities within drugs. Counterfeit medicine can contain the same impurities as genuine products, which means that relying solely on routine HPLC testing commonly used for the battery list may fail to detect them. This neglect the risk of overlooking counterfeit drugs and emphasize the importance of analyzing the interformulation for accurate authentication. Raman spectroscopy is referred to as one of the techniques commonly used for rapid and non-destructive analysis of counterfeit tablets. Raman spectroscopy was initially covered because of its advantage as a fast, non-destructive technique that does not require sample preparation. This technique has been widely used in previous studies to distinguish between genuine and counterfeit drugs by detecting difference in chemical composition and molecular structure. When used on the counterfeit psychotropic tablets in the study we referred to, Raman was unable to reveal significant difference between genuine and counterfeit samples. This is likely due to the close similarity of physical and chemical formulation or interference for fluorescence that disrupt the Raman spectrum. Although Raman spectroscopy is theoretically effective for screening counterfeit drugs, Raman is not always suitable for ARCAS, especially when the counterfeit is done with a chemical composition that is very similar to the original drug. This shows the importance of a multi-technique strategy in forensic drug analysis, and not relying on a single method. This is the graphic from the discussion we've been tackling before.
Regardless their chemical compositions, counterfeit medicines pose significant risks to the patients due to their unknown and unregulated manufacturing and storage conditions. While some counterfeits lack active ingredients, others closely mimic genuine products, making detection more challenging. Therefore, a comprehensive analytical strategy is essential to identify all types of counterfeits, whether they contain the correct active substance or not. Involvement of analysis of counterfeit psychotropic drugs can be done through various methods, including chromatography, spectroscopy, imaging, and elemental analysis. A thorough examination of excipient formulations is crucial to ensure the counterfeit goes undetected. Using a combination of analytical techniques allows for a complete chemical profile once a product is confirmed to be fake. Spectroscopic tools in particular are available for quick and non-destructive screening, enabling direct comparisons of the overall compositions between counterfeit and authentic medicines. The data obtained from this analysis are not only vital for quality control, but also serve a broader purpose in forensic investigations. They support pharmaceutical companies in detecting counterfeit sources and contribute to prevention strategies, reinforcing efforts to protect public health and maintain the integrity of the drug supply chain.
Last but not least, this is the case reference we used on this video. So, thank you for watching.